Dear students, our today's topic is endosperm, its types and development. In this topic, we will discuss what is endosperm, how it is formed, what are types of endosperm, and what is the function of endosperm. First of all, we will try to understand what is endosperm. Endosperm is actually primary storage tissue which is present in the developing seed. It is one of the most vital part of the fertilized embryo. Vital part is because it provides nourishment to the developing embryo. Without endosperm, embryo cannot develop into new seedling or a new plant. Endosperm structure is characteristic of angiosperms. That is, it is found only in angiosperm group of plants. Other groups of plants like algae, fungi, bryophyta, pteridophyta, and gymnosperms, they lack this structure. Now, how endosperm is formed? Endosperm is formed as a result of double fertilization, which is unique characteristic of angiosperm. Double fertilization means that one male nucleus fuses with the egg to form zygote, which ultimately develop into embryo. And the second male gamete fuses with the secondary nucleus or the polar nuclei to form primary endosperm nucleus. This fusion is also called as triple fusion because here three nuclei fuse together, one of male nucleus and other two polar nuclei, which result in formation of a triplet structure that is called as endosperm and which provide nutrition to the embryo. Now, endosperms are classified or distinguished into three types. One is nuclear endosperm, second cellular endosperm, third halobial endosperm. These are classified or differentiated on the basis of their development. Now, first, in case of nuclear endosperm, here how it get develops. Now, this is characterized by free nuclear division. Free nuclear division means that only nuclei divide. One nucleus divides into two, two into four, four into eight, and so on. In this way, thousands of nuclei are formed. All these nuclei then come to lie on periphery, and there is formation of a central vacuole, as is clear in picture. After completion of free nuclear division, then cytoplasmic division occurs. And cytoplasmic division occurs from periphery to center, which ultimately result in formation of a cellular tissue, which is called endosperm. Because development is through free nuclear division, so it is called as free nuclear endosperm. The most common example is coconut. As we see in young coconut, we will find coconut water or coconut milk, which is actually free nuclear endosperm. As the coconut matures and cytoplasmic division occurs, we will find that there is endosperm, which is cellular in nature. Second type is cellular endosperm. Here, as name indicate, nuclear division is followed by cytoplasmic division. As we see in the diagram, when one nucleus divides into two, then cytoplasmic division occurs and there is formation of two cells. From two cells, there is formation of four cells, from 4 to 8, and so on. And ultimately, there is formation of large number of cells which form the cellular endosperm. Third category is fallopian endosperm, which is actually in between nuclear and cellular endosperm. Here occurs both type of development, means first division is cytoplasmic division. The nucleus divides into two and followed by cytoplasmic division, which result in formation of two cells. One larger towards the micropyle end, that is called as micropylar chamber, and one small toward the chalazal end, that is called a chalazal chamber. Now, as is clear from the diagram, in micropylar end, there occurs free nuclear division as in first case. Okay, nuclei divide and redivide, but it is not followed by cytoplasmic division. And ultimately, there is formation of large number of free nuclei, and then cytoplasmic division occurs from periphery to center, as in case of nuclear endosperm. While in the lower part, that is toward the chalazal chamber, there occurs cellular division. Each nucleus division is followed by cytoplasmic division and ultimately result in formation of large number of cells. But ultimately, the whole endosperm become cellular. So it is haloidal type because it shows both free nuclear division as well as cellular type division. Now in this slide you can see the detail of this nuclear endosperm, cellular endosperm. Their example in nuclear endosperm, the common example are coconut, capsella, arachis, and in cellular endosperm, the common example are adoxa, helianthus, etc. And in halobial, there is example of hydrilla and Balisneria. Now another important feature is, as I've already told you, that the function of endosperm is provide nourishment to the developing embryo. Now during development of embryo in some seeds, the endosperm is completely consumed in its development, and when the mature seed is formed, there is no endosperm remains. Such seeds which are without endosperm, they are called as non-endospermic seeds or ex-albuminous seeds. Its common examples are dicot seeds like pea, groundnut, and in other case like monocots. Here, when embryo develops into new seedling, it consumes the endosperm, but it partly consumes, and a part of endosperm remains in the mature seed. So seed will have endosperm, and such seeds are called as endospermic seeds or aluminous seeds. For example, coconut, maize, rice, etc. Now functions of endosperm. The major and most important function of endosperm is provide nourishment to the developing embryo. That is why it is commonly called as nutritive tissue that provides nutrition to the embryo. By taking nutrition from the endosperm, this embryo can develop into a mature seedling. And second function is endosperm, it regulates the precise mode of embryo development. It helps in proper development of the embryo. So this is all about the endosperm. I hope you will benefit from it, and if there is any query, then please you can send your query to our WhatsApp group. Thank you.